So, good afternoon, everybody. My name is Professor Lindsay Hall, and I'm the Chair of Intestinal Microbiome at the Technical University of Munich, but I also hold a position at the Quadram Institute Bioscience in the UK in Norwich. And first of all, I'd like to say thank you very, very much for the kind invitation to give a talk at the 65th anniversary of the Faculty of Microbiology at the University of Costa Rica. And what I'm going to do for the next 45 minutes is just give you an insight into some of the work that we're doing, which is primarily focused on the gut microbiota and specifically in the early life developmental window. And if you want to find out a little bit more about what we do, I'm not going to cover obviously everything in this 45 minutes. We have a website. But we basically have three main research avenues in the team. So we look at micro-post crosstalk, we also look at diet microbiota interactions, and we're also very interested in infection resistance and rapid diagnostics as well. So I will touch upon all of these points as we go through, but we obviously have some other research interests as well. So let's dive right into it. So as I've mentioned, we're particularly interested in the team at looking at the neonatal gut microbiota. So those first hours, days and weeks post-birth is very important for establishing the microbial ecosystem. So these microbes, we're not just, a house for our microbes, and we don't just carry them around. They provide us with a huge range of beneficial functions, including improving the gut barrier, so strengthening that barrier, which is very important for our overall health. They help program the immune system both locally in the gut, but also systemically as well. And of course, they're incredibly important for digesting the food that we eat as well, because there's many different food components that we eat that we can't actually digest ourselves, but our microbes that reside in the gut do that job for us. So specifically thinking about those first stages of life and when our microbial ecosystem is first being established, and this is just a recent review that I did with one of the postdocs in the team, Matthew, last year, that was published in F1000 Research, which just looks at some of the, the brand new research in this area. So we have a relatively, unstable ecosystem in the first even hours, days, and even weeks post-birth that's incredibly susceptible to different external factors that may change the gut microbes that are found present in the gastrointestinal tract. And we're predominantly vaginally delivered breastfed babies are dominated with bifidobacterium, which is going to be one of the main microbes I talk about as we move through the presentation. But there's other microbes that are very important also in these first stages of life. But I think what's important to highlight here, because we'll talk about this later, is the fact that certain factors like how a baby's born, if mum or baby have had a course of antibiotics, and how the baby is fed, so breastfeeding or formula feeding or a mixture of both, this can radically disrupt the gut microbiota at these first stages. And this is really important because the ecosystem here is very fragile because it's not fully established yet. So it's very important to understand how some of these factors may negatively influence the gut microbiota and what that might mean for child health. So as I've mentioned, one of our favorite bacteria that we work on in the group is bifidobacterium. And we're particularly interested in this bacterial genus because we see them as microbial pioneers or microbial founder species. And we've known for many, many years that overabundance of this particular taxa is also associated with improved health in infants. And if we just talk about this microbe in a bit more detail here, so BIF are really one of these first microbes that colonize the infant post-birth. And in fact, that seems to be from a number of different places. So bifidobacterium is actually found in the maternal gut, and then you get transfer from the maternal gut during childbirth to the infant. The vaginal microbiome may also have bifidobacterium that's passed on during natural childbirth. And also there's potentially bifidobacterium in breast milk as well, which is another way for the mum to seed her baby with bifidobacterium. So they're found at incredibly high levels in these first stages of life. And these high levels do correlate with improved health and actually reduced numbers of hospital visits for these infants. But what's important to say here is that there's a really strong link with the levels of bifidobacterium and diet. So breastfed babies, basically the ecosystem is dominated with this bacteriotaxus. About 80 to 90% of the total community is bifidobacterium. But in formula-fed babies, they do have bifidobacterium, but the relative abundance or even absolute abundance is much lower. And we'll talk about why we have that association kind of in a moment. I think what's important here is that I think everybody knows that bifidobacterium is one of the main groups that are used in probiotics, the ones, you know, you can obviously buy in the pharmacy or kind of in the supermarket. And although kind of there's not really any health claims substantiated for using these food supplements, I think what's important to say, and this is one of the reasons why we're working on this microbe, is the fact that they're GRAS microorganisms, so they're generally recognized as safe, which does mean if we have something that we want to move into the clinic or we want to move into humans, we can do this very quickly because of the strong safety track record. But I think the other thing that I want to highlight here, and I mentioned it on the previous slide, is these the early life microbiota and bifidobacterium in particular are incredibly susceptible to some of the external factors that these microbes come up against. So including antibiotics and diet. And again, I'll touch upon these as we move through. But let's kind of just start talking a little bit more about the fact that I said about the very early life microbiota. And this means that we're going to be talking about premature babies. So premature babies, and this is kind of in the UK or Europe, but I appreciate this definition changes depending on where you are globally, but kind of generally preterm babies are those babies born less than 37 weeks gestation. They're often very low birth weight, so about 1.5 kilograms, and full-term babies are about 3.5 or 4 kilograms. And at the moment, about 1 in 9 live births globally are defined as preterm, and that number is actually rising. So these are a very important group of infants to look at. Because these babies are born prematurely, they have an underdeveloped immune system in comparison to their full-term counterparts. And they're also exposed to some of these microbiota modulating factors I mentioned. Many of these babies are born by cesarean section. They reside in sterile neonatal intensive care units, which are obviously critical for keeping these fragile babies alive. And in many, many cases, these babies get a huge amount of antibiotics, including prophylactic antibiotics. In the UK, for example, if a baby is born less than 1.5 kilograms, they're automatically prescribed benzyl penicillin and gentamicin for kind of a period of 3 to 6 days. So in fact, it's actually very difficult normally to find a preterm baby that has not had a course of antibiotics. So these units and the clinicians and the healthcare professionals that work in them are absolutely critical for keeping these fragile babies alive. But the preterm microbiota looks radically different from full-term vaginally delivered babies. They have markedly reduced levels of bifidobacterium, and worryingly, they actually have really high levels of potential pathogens in there as well. So kind of E. coli, Klebsiella, Enterococcus, and actually most of these are normally hospital acquired as well. So as you can imagine, are potentially posing a health threat to these fragile babies. So just to talk a little bit more in detail about the preterm gut and why potentially it provides a niche for overgrowth of particular pathogens, and I'll talk about a specific disease in a moment, is that the gut barrier isn't formed properly, as I already mentioned. They've got an immature immune system, and they've normally got a thinner mucus layer as well, which means that pathogens might have more access to the epithelial barrier, so the single cell layer that lines the gastrointestinal tract. Because they lack key microbiota members like bifidobacterium, there's kind of less competition for nutrients, for kind of niches within the gastrointestinal tract. And also some of these microbes also produce novel antimicrobials, including bacteriocins, that actually kill potentially pathogenic microbes. So the preterm gut is almost an ideal environment for overgrowth of particularly pathogenic microbes that might be associated with diseases. And one of the diseases that it is associated with is necrotizing enterocolitis. And this is actually the most common gastrointestinal emergency kind of worldwide in neonatal intensive care units. It's got a huge impact in terms of mortality, which is up to 40%. And if a baby does survive from NEC, they often have longer term health complications, including kind of cognitive development and also immune pathologies later as well. And I think, as I've already mentioned, NEC is one of those diseases that's linked to growth of pathogens that are already maybe in the gut or have come in from elsewhere from the hospital environment, for example. And these are just some of the kind of, I guess, clinical characteristics of necrotizing enterocolitis. So what's important to say here is the baby will look okay. And then the doctor or the nurse will go around and do their ward rounds, and then they will basically see the baby presenting maybe with a distended abdomen. They might get rushed off for x-ray where they kind of present with this pneumonitis intestinalis, which is gas cysts in the bowel wall. And then if the baby is very ill, they will be rushed into surgery and they will have the necrotic part of the bowel, which you can see here, actually removed by neonatal surgeons and then obviously kind of brought back into recovery. But they've obviously had a large part of the bowel removed, which is really not ideal for a preterm infant. So this is kind of where we stepped in and where we first started working on this project, which was a couple of years ago now, which is kind of the microbiota is not right in these preterm babies. So can we rebalance the microbiota and reduce the incidence of diseases like necrotizing enterocolitis? So this is just introducing our preterm cohort study called BAMBI, so baby-associated microbiota of the intestine. And this was a multi-center study that covered 4 different hospitals in England. And we had 2 different groups. We had babies that are routine supplemented with probiotics, and the probiotic is here, it's in Floran, which has Bifidobacterium bifidum and Lactobacillus acidophilus. And then we had other babies that were routinely not receiving probiotics. We took a number of samples when they were in the neonatal intensive care units, and then we basically started to look at their microbiome over time and to correlate that back to kind of health outcomes. So just as a kind of first overview of some of the information you get when you do this kind of microbiome profiling studies from stool samples or poo samples, is this is kind of the first time point we looked at in the study. So kind of really the first kind of week post-birth. And remember, this is our control babies that don't have probiotics and our biflacto babies that are receiving probiotics. And what's important to say here, this is an NMDS plot, which is basically an unbiased way at looking at your microbiome data. And what you can see very clearly is there's already a really quite significant divide between the red squares, which are control babies, and our blue triangles, which are supplemented babies. And if we drill down to the data a little bit more, we can see that Bifidobacterium appears to be driving the separation of the biflacto supplemented babies, whereas we've got potential pathogens including Staphylococcus, Escherichia, and Klebsiella that appear to be driving the separation in the control babies. But let's look at this kind of across the time points, and we can see that really nice divide between our 2 groups right across the study period. But if we delve into this a little bit more, and this is kind of just using a heat map to kind of show the data in a slightly different way, is that we can see our babies normally cluster into kind of 6 main bacterial taxa. So the babies are normally dominated with one particular group. And this is, again, the first kind of age, but we see this kind of changing over time a little bit, particularly in the probiotic supplemented group, but I'll show that now in a moment. So this kind of shows you a bit more in a bit more detail. So each of these points are individual babies, and this is just the first time point, again, post-birth. So what we can see very clearly that in the first kind of time point, we actually see that there's Staphylococcus is actually high in both of our baby groups, whereas Bifidobacterium really is absolutely dominating only our supplemented babies. And we're seeing kind of dominance of potential pathogens in our control babies, but lower relative abundance of these potential pathogens in our control babies. So E. coli, Kleb, Streptococcus, and Enterobacter as well. And we see very similar profiles for this right across the study period, although we do start to see Staphylococcus decreasing in our supplemented babies. But let's kind of look at the Bifidobacterium in a little more detail, because this is the one that we're interested in, and we want to see what's happening here in a bit more detail. So this is just to kind of show you longitudinally. You can see that the supplemented babies have more Bifidobacterium right across the study period, and you can actually see that that goes up. What's important to say, in the first kind of 2 time points, the control babies, only a handful of them have Bifidobacterium within their gut, but actually they appear to get naturally colonized the longer they're in the neonatal intensive care unit. But what's important to say here is that this is obviously a delayed colonization, and that has implications for immune programming. So even though they get BIF, it might be too late to kind of basically beneficially program both the mucosal and systemic immune systems. And I mentioned some of the external factors that are really important in kind of, I guess, negatively impacting the early life gut microbiota. So I mentioned about delivery mode. What's actually important here is that actually bifidobacterium relative abundance is not impacted if a baby is born by cesarean section or if a baby is born vaginally. But this is probably due to the fact that these babies receive this prophylactic antibiotics. So even if there was any BIF transferred during natural childbirth, they basically would be wiped out because an antibiotic course has come into the babies. And unsurprisingly, bifidobacterium are not one of these multi-drug resistant kind of bacterial attacks that we really need to worry about in the kind of global threat of antimicrobial resistance. They're very susceptible to antibiotics. So what we see here is that if a baby's on a course of antibiotics, they always have lower abundances of bifidobacterium. However, if we look at pathogens that normally are multi-drug resistant, like E. coli, Klebsiella, and Staphylococcus, the relative abundance of these taxa are not impacted by antibiotics, which again indicate that they are multi-drug resistant, and I'll show you some data later to indicate that that is in fact the case. The other thing that's really important here from a clinical perspective is the lower birth weight a baby is, and kind of the smaller a baby is at gestational kind of birth, they also have less bifidobacterium. And this is important because these are the babies that are normally at higher risk of necrotizing enterocolitis. So we need to think of other ways to really get optimal bifidobacterium colonization in these fragile, very premature, very low birth weight babies. So what we kind of know, I guess, from many other probiotic studies, which kind of in most cases have been carried out in adults or in full-term babies, is that we normally see we need to give kind of daily supplementation, otherwise the probiotic strain just gets washed out the other end because they're not very efficient at colonizing. But what's interesting from our Bambi studies, we actually saw persistence of bifidobacterium in the preterm gut. And that's just important to show here, is that the babies stopped receiving the probiotic supplementation after they'd reached post-conceptual age of 34 weeks. But we obviously collected samples after this time point. And interestingly, we still see bifidobacterium relative abundance being high. And if we look at this in a bit more detail, not just looking at genus level, but species level, this becomes very clear. So if you remember, it's Bifidobacterium bifidum, which is the species found in the probiotic. And I think this is a really nice graph. You can see the supplemented babies are chock-a-block full of this particular BIF species. We do not see this in the control babies, who, if you remember, do start to get bifidobacterium, but this is like B. longum and B. brevi, not B. bifidum. And then what we did to confirm, because this is obviously just done on a kind of 16S microbiota profiling, is that we also did isolation of bifidobacterium and whole genome sequencing. And from these isolates that were collected from babies after the supplementation had stopped, we sequenced their whole genome and we basically aligned that back to the reference strain. And in this case, the reference strain was the Bifidobacterium bifidum infloran strain. And what we can see here is that there's basically no single nucleotide polymorphisms difference. They're basically identical, which gives us confidence to say that it's not just any Bifidobacterium bifidum in these supplemented babies, it's actually the probiotic strain, which I think is really important to say. And because we had the genome sequence of our infloran strain, and one of the things I mentioned in terms of diet, kind of breastfeeding or formula feeding, is that Bifidobacterium is one of these kind of groups of bacteria that we know is really linked to the infant diet. So they often encode enzymes in their kind of genome that are available to degrade some of the complex sugars found in breast milk called human milk oligosaccharides. So if we look for the gene sequence of these enzymatic clusters, we can find them. So these are just suggesting kind of some of the other genes that are associated with digestion of these complex sugars, these HMOs. So we can look at the gene sequence, but of course having the presence of a gene doesn't mean you have a phenotype. But actually what we can see here is that if we look at our infloran isolate, it loves growing in whole breast milk. But actually, if we also look at the growth, but with specific human milk oligosaccharides like 2FL and LNNT, we can also see that they grow very, very well, which correlates really nicely back with that genomic signature. So the Bifidobacterium and florin strain can utilize the diet of these babies. And what I should have said, which I haven't already said, and apologies, is that nearly all of our preterm babies received either express breast milk from mum or donor breast milk. We had a very small number on formula. So really this is the perfect diet for this infloran strain. We had done 16S ribosomal RNA microbiota profiling, but we were really interested to look into kind of, I guess, the really functional, outputs of this probiotic supplementation. And this was in collaboration with John and Femina down at Imperial College London and the University of Southampton. So this is just kind of this is an NMR plot here, kind of showing some of the metabolites that we found in our preterm babies. But I guess the take-home message here is that our BIF lactose supplemented babies had far higher acetate, which you can see here by the big massive peak. And they also had higher lactate as well. And also Christina, who was one of the kind of the lead people on this study, who was a PhD student at the time, she actually went and measured the pH of the fecal samples, and we saw a really beautiful correlation between presence of these short chain fatty acids, including acetate and lactate, and a reduced pH. So preterm babies that are being supplemented had a stool pH of 5.5 comparison to pH 7, which is what we saw in our control babies or our non-supplemented babies. And this is important because many pathogenic microbes don't like lower pH, they don't grow as well. So we think this is a really important colonization resistance mechanism. And then if we correlate the metabolites we profiled with our microbiome profiling, and this is just a Spearman correlation map, you don't need to look at everything here, just maybe look at the bifidobacterium here at the bottom, is that we do see a direct correlation between levels of bifidobacterium and acetate and lactate. And interestingly, we see lower amounts of breast milk sugars, so 2FL and 3FL. So what's happening here? Well, we already showed this in the growth curves that we did. So the presence of bifidobacterium in the preterm gut is they're fermenting the human milk oligosaccharides in the breast milk. The fermentation end products of this metabolism are lactate and acetate, and that links to the decrease in pH. So we've given the right diet, the right microbes, in this case Bifidobacterium bifidum, are fermenting them, and then we're getting an actual whole kind of change in the chemical composition of the gut, which is probably then linking back to the fact that we have far reduced abundance of potential pathogens in these preterm babies. So we've done the microbiota profiling, but of course from a clinical perspective, who cares? So we changed the microbiome, but does this make an impact onto babies' health and well-being? Well, I'm delighted to say that this was the case. So this was an incredible kind of clinical study led by Claire Robertson, who was a trainee clinician in the lab, and Paul Clark, who's our consultant neonatologist that we work with on many different studies. And what we found was that the probiotic supplementation actually reduced the incidence of necrotizing enterocolitis by over 50%. And this was also the case for late onset sepsis as well. And in fact, all cases of mortality. So this is, great to see a change in the microbiome, which actually also impacts, we're actually having more babies surviving, which is, obviously fantastic news. So we're just kind of following up on this clinically at the moment. So just to kind of summarize this kind of first part of the talk, is that normally preterm babies are dominated by potential pathogenic microbes, which might be linked to necrotizing enterocolitis. However, if we give them a probiotic supplementation, and in this case, it was with the Bifidobacterium bifidum and the Lactobacillus acidophilus. I didn't talk about lacto, that might be a question that somebody wants to ask me, is that we see Bifidobacterium colonization, which is very effective, and we see a concurrent reduction in potential pathogenic microbes linked to necrotizing enterocolitis. We do see Lactobacillus colonizing some babies, but this is only a very small number. We see that Bifidobacterium abundance is actually negatively impacted by if the baby's lower birth weight and if the baby's had a course of antibiotics, and this might be basically linked to the niche that's available for Bifidobacterium to colonize. But of course, clinically, we saw this incredible 50% reduction in NEC, which was kind of really, amazing to have that mechanistic lab work, and then linking that back to the clinic and from the translational perspective. So at the moment, what we're trying to do with these data is really highlight the important role that bifidobacterium plays in the preterm gut. But what's important to say is that you also need to give the right diet so you get efficient colonization. And in this case, it's breast milk and these human milk oligosaccharides. So there's been actually a number of different neonatal intensive care units that have started to use probiotics routinely in the UK on the back of this kind of work that we've done, which is really great to see the impact. So that's kind of the first part. We're now going to change track a little bit. And we're actually going to think about necrotizing enterocolitis as a disease that's associated with overgrowth of pathogens, but it's associated with a very rapid deterioration clinically. And some of the pathogens that have been associated with necrotizing enterocolitis include E. coli, group B streptococcus, Klebsiella, Enterobacter, and also Clostridium perfringens, which I'll talk about at the end. And I think everyone probably knows that's listening is that nearly all of these microbes are associated with being multi-drug resistant. So we've got a huge problem here. We've got these babies that have overgrowth of pathogens. They go downhill very quickly. So we need to come up with a way to potentially rapidly kind of profile the gut microbiota and also to look for antibiotic resistance profiles. And this is kind of where we've been leveraging some of the newer sequencing technologies that are on the market. And this is the Oxford Nanopore MinION. And this is very attractive from a in-field or clinical deployment perspective. It's very small, which is very handy. But I think what's most handy is the fact that you can prepare the samples very quickly and you can get real-time sequencing information immediately when you load your sample onto the MinION. And the other advantage of this as a methodology is that we can do shotgun metagenomics. So instead of just looking at kind of the genus of the bacteria, which is not very useful from a diagnostic perspective, we can get down to species level, if not strain level. We can look for actual abundances rather than relative abundances, which is again very important to make sure we're seeing potential overgrowth. And the other thing that's really important, because we're obviously sequencing all of the DNA in the sample, not like a marker gene like 16S ribosomal RNA, we can look for key genes, and in this perspective, antimicrobial resistance genes. So this was a phenomenal kind of collaboration with some of our colleagues at the Earlham Institute, which is just across the road from the Quadram Institute, also in Norwich in the UK. And this was with Darren, Matt, and Richard, and Christina was also involved in this study as well. So we did due diligence and we did a lot of benchmarking before we started using clinical samples, kind of using mock communities. And kind of based on that data that we got, we were then confident enough to start using kind of precious clinical samples to see if we could kind of optimize the MinION for rapid diagnostics or rapid microbiome profiling. And this is just a PCOA plot that is just kind of indicating some of the samples that we used in study and also showing the very different microbiome profiles we get from our preterm babies. So we included healthy preterm babies, which were dominated with bifidobacterium, but we also had babies that had developed necrotizing enterocolitis or sepsis. And obviously these were the babies we were interested from a diagnostic perspective. So we first did kind of benchmarking with clinical samples as we took a number of our babies from the Bambi study. And for this particular one, this was baby P10, so 26 weeker, just over a kilogram in weight. So kind of, pretty preterm and also low birth weight. You can see here that the babies also received a number of different antibiotics, and they've also had 2 episodes of sepsis as well. And what we did here is we looked at kind of the bacterial taxa information we were getting, both from our usual Illumina sequencing that we would do, which is kind of the gold standard, versus what we were getting from the nanopore runs. And we basically see a beautiful direct correlation, which really gives us confidence that what we're seeing with the MinION is incredibly similar to what we're seeing with Illumina, which gives us confidence moving forward with this pipeline. So we can just look at this in a bit more detail using these pie charts here. These are the 3 different time points with baby P10. We can see there's an overabundance of Klebsiella pneumoniae, then it's Enterobacter cloacae, and then we're back to kind of Klebsiella again. But again, we can see very similar profiles between the 2 different methods. And the other thing we also had a look at was the overall composition of the antibiotic resistance genes in the total microbiome community, so the so-called resistome. And you can see quite terrifyingly here, we see a huge number of potential antibiotic resistance genes, but what's important is we see a very similar profile between the Illumina and the Oxford nanopore sequencing data as well. So if we just have a look at one of our other babies, and this is P205, 22 weeker and just over 500 grams. And this is actually one of the babies that develop necrotizing enterocolitis. Huge amounts of antibiotics here that are being hit, that are hitting the preterm microbiome. What we did here, and this was kind of why it was so fantastic to work with kind of Richard and Matt and Darren, is that Richard's kind of a software developer, he's a bioinformatician. And he kind of added on bespoke software that enabled us to rapidly kind of identify what microbes were present in the preterm gut over time. So in this particular baby, what we see as quickly as 1 hour sequencing start, we see it's dominated by Enterobacter cloacae. And then if we start looking at the antibiotic resistance profiles in the preterm gut of this baby, we can start seeing a whole load of antibiotic resistance classes popping up. And maybe unsurprisingly here, the longer we sequence, the more sequencing depth we get. So then we start to see kind of, I guess, those antibiotic resistance determinants that are found at lower abundance. But we can, I think it's a lovely way of showing it, but it also shows that this baby is, again, I guess, completely full of potential pathogenic microbes linked to the necrotizing enterocolitis that we saw that are also potentially multi-drug resistant. However, this is the resistome. So these are all the antibiotic resistance genes found in the whole microbiome. But clinically, that's not very helpful. What we really need to know is what microbe is carrying what antibiotic resistance determinant, okay? So again, with Richard, what we did was we added some functionality to the software that he'd already developed, so nano-OK RT, so real time, and we added what we called walkout analysis. So what that does is when basically we detect an antibiotic resistance gene, we then look 50 base pairs either side of that gene sequence, and then we use that information to basically ID the bacteria that's carrying it. And from this, what we could see from our previous baby, that maybe unsurprisingly, that we saw a lot of the antibiotic resistance genes were mapping back to Enterobacter cloacae, which is one of these pathogens that have been associated with NEC. And in this case, it's a huge problem because they're also multi-drug resistant. Now, all of that we did in an academic lab, and we were like, we were obviously really robust. We took our time, but we got to the point where we realized that it would be important to determine how fast can we go, right? So how quickly can we go from a stool sample to having clinically actionable data that doctors and healthcare professionals can actually use for treatment of these fragile babies? So we did this with one of our babies, P8, and this is kind of just the timeline of what we did here. So we obviously took a sample collection, we did DNA extraction, we prepared the libraries, we did DNA quality control, and then we basically loaded it onto the MinION, and then we got real-time data out the other end. And I think what's again kind of as on the previous slide, that even after 1 hour sequencing start, in this case, this baby's beyond dominated with Klebsiella pneumoniae, but it doesn't matter if we have another 5 hours sequencing, we've got the same profile. So we can basically say Klebsiella pneumoniae 1 hour after sequencing start. And then again, we can look at the total resistome, and we see a whole load of antibiotic classes beginning to pop up. And of course, some of these we see sooner rather than others because they're found at higher kind of absolute abundance. And when we did the walkout analysis on this particular baby, we found that about 75% of the antimicrobial resistance genes were mapping back to Klebsiella. And Klebsiella is a real concern because of the emergence of multi-drug resistant clones that obviously basically are able to survive against numerous different antibiotics, but then potentially could also share these antibiotic resistance genes through horizontal gene transfer with other pathogens in the preterm gut as well. So we also did kind of gold standard microbiology testing, where we isolated the Klebsiella from baby P8, and we did whole genome sequencing. And what was important here was we were able to basically map the whole genome, which of course is again the gold standard with the shotgun metagenomic data, and we show a really beautiful correlation between those two different methods, which was great. We also found a number of different antibiotic resistance determinants. And then when we did minimum inhibitory concentration assays to actually look for phenotypic antibiotic resistance, this linked up very nicely with the genotypic resistance profiles we got from the MinION run, which was fantastic. So we kind of show here there's a validation. And we also did a whole load of mock community work on here to make sure we were seeing what we would expect. So just to kind of summarize, this part of the talk is that, leveraging nanopore, Oxford nanopore in the MinION means that we can rapidly profile the preterm gut over time using shotgun metagenomics, kind of looking for episodes of sepsis, or indeed data I didn't show. We can also look for probiotic colonization with bifidobacterium. We can do this in real time, so we can go from basically the clinical sample all the way to providing actionable data that a clinician can use. And actually what we did here is we worked with neonatologists and gave them the profiles that we got, and they would have actually changed the antibiotics that this baby had received if they'd had this data. And we did this kind of through a number of different kind of software developments, which were kind of led by Richard and the team. We did a whole amount of validation. Data I didn't show you, we went from the MinION to the Flongle, which is even smaller, but most importantly, cheaper, which makes it more attractive from a clinical perspective. And actually Richard and his PhD student Ned have been working really hard on this, and they've actually just released new software for doing the beautiful dashboard and looking at shotgun metagenomics as well. And I think at the moment, we're just kind of seeing how we can potentially move forward with this, because of course this is very small, this is a pilot, we're kind of only doing validation. We'd like to move forward into the clinic, do this on a far bigger scale, and actually do this in a way that can be used by those like treating these babies in neonatal intensive care units, again, with the idea of reducing the incidence of necrotizing enterocolitis. Now, we will go on to nearly the final part of my talk, which is a little pathogen I mentioned earlier called Clostridium perfringens. So Clostridium perfringens is one of these microbes that I remember from my undergraduate microbiology degree that causes gas gangrene. But actually, if you read the literature, this particular pathogen is associated with many different enteric diseases, so gut diseases. And in fact, this is normally the case in kind of neonatal animals where it's a huge problem. But more recently, because people have started looking at the preterm gut microbiota, it actually appears that Clostridium perfringens is also associated with necrotizing enterocolitis, but very specific necrotizing enterocolitis. So Clostridium perfringens associated necrotizing enterocolitis, which is actually normally found to be clinically much more devastating, with actually a far higher mortality rate as well. And this kind of Simon Kroll and his team down at Imperial College London are kind of been the ones that have been driving this. So this is a beautiful collaboration we established with them over the last couple of years. And this is just some of the babies from some of their studies they've done. And the red here is the Clostridium. And what you can see is so this is a longitudinal profile of the microbiome. And the red is Clostridium perfringens. And what you can see is that we see a steady increase in this pathogen just prior to NEC onset. And in fact, people have done this in animal models and actually kind of obviously done Cox postulates to show that perfringens is a causative agent of preterm necrotizing enterocolitis. So, but Clostridium perfringens can also be found in, I guess, healthy preterm babies. So what we want to do here, and we want to do this really rigorously. So we basically went and isolated Clostridium perfringens from a huge number of poo samples from preterm babies. And we did whole genome sequencing. And we were looking for genomic signatures to see if there was a NEC signature versus maybe more of a commensal Clostridium perfringens that didn't have maybe specific virulence factors. So this was led by the fabulous Raymond, who was a PhD student in the lab and he's now a postdoc. And Raymond, as I said, was really interested to see if there was a genomic signature between, I guess, preterm Clostridium perfringens strains and those that had NEC versus those that didn't. So this is actually part of a far bigger study. It's unpublished. We're hopefully going to be maybe pre-printing by the end of the month, so you can look at all the detail then. About 300 Clostridium perfringens strains isolated from preterm babies. Raymond obviously did a phylogenetic analysis to look at the evolutionary relationships, and he also looked for, kind of important virulence factors like toxin genes, colonization factors, and antimicrobial resistance profiles. And what's important to say here is that we almost had kind of a lineage that's down the bottom here, lineage 4, And this comprises of Clostridium perfringens strains that are not from neck babies. And they basically, if you add up all the virulence genes, they have far lower. And if we look at this in a bit more detail, I think this is really interesting, is that commensal Clostridium perfringens do not encode PFOA, which is perfringilysin O. And this is a pore-forming toxin that's encoded on the chromosome. And you can see this clearly here from when Raymond's done the counts. And actually, we've actually done a whole load of work, but I'm only going to show a snapshot here, is that if you take a strain that encodes this toxin, so from a neck Clostridium perfringens strain, and you compare that to commensal Clostridium perfringens strain that doesn't encode the toxin, it correlates beautifully with necrosis using epithelial cell lines in vitro. And you can see this very clearly here. If PFOA is encoded, you get really high necrosis, and you don't if it's not encoded. So that's kind of nice because that links really well with pathogenesis or the pathology of neck. We also see more colonization factors being encoded in the neck strains of Clostridium perfringens. Of course, you know, a strain's got to get in, but it's got to colonize if it's going to be initiating disease. And we also looked at other colonization factors as well. And we saw that, you know, the commensal Clostridium perfringens that aren't associated with neck versus strains that are, and we saw that difference in these virulence factors. And then, of course, because we have whole genome sequence data and we've got longitudinal samples, we can do super fun genomic epidemiology. And that's what we did, because when we drilled down into one of our particular kind of clades that had neck associated strains, we basically had 31 strains that were only 4 nucleotide polymorphisms different from 6 preterm babies, so they're clonal. And then Raymond did a whole load of analysis, and we looked back at the clinical metadata and how these babies were transferred. And basically what we were able to show was a transfer kind of within babies, within a ward of a hospital, and also between hospital. So in hospital 1, we see baby N43 with this particular strain. It then passes this on to twins N45 and N46. These twins are transferred to hospital 2. Then we see the strain popping up in other preterm babies within this new hospital, who then pass it on to baby N20. And actually, this baby at the end actually ultimately succumbed to necrotizing enterocolitis. So again, highlighting how important it is to track these strains and think about prevention measures. So we think perfringens is a really important pathogen in actually preterm necrotizing enterocolitis. And we've got these, I guess, Clostridium perfringens strains that are hypovirulent, so kind of a normal member of the preterm microbiota, don't cause a problem, versus strains that are much more hypervirulent, encoding virulence factors like toxins, colonization factors that might initiate pathogenesis in neck. We can see that these strains are transferred kind of between babies, between hospitals, and actually some of these strains persist for an incredibly long period of time, because we've looked at babies 2 years later and they've still got the same strain. And I think we're kind of we want to use this evidence to indicate that perfringens is probably one of these strains that's understudied at the moment in the context of preterm neck. And we really need to be thinking about this at the moment. So that's kind of a wrap up of some of the studies that we're doing on the very early gut microbiota. And I guess the good, we have to go back to him, the bad, thinking about the diagnostics and E. coli and Klebsiella, and the downright ugly with respect to Clostridium perfringens and Clostridium perfringens associated with neck. But I just wanted in the last kind of 2 slides just to highlight some of the really exciting work that we've now got going with people in Costa Rica. So a Costa Rican twist, and thinking again about Clostridium perfringens as being a really important but understudied pathogen. And this is with the fabulous Cesar Rodriguez. So I'm just going to show you some of the data that we've generated, and you can also ask me questions, but you can also run and ask Cesar questions as well. And again, what we've done here is that we've been working with Cesar and his team to look at Clostridium perfringens that are found in Costa Rica from both animals and humans in different types of diseases. And we've done whole genome sequencing, and we've compared this to some of the other strains that we have in our collection, and also from strains that are found from all over the world. So I don't expect you to kind of look at this all in a lot of detail, because this is very preliminary analysis, but we see a range of different toxin genes that are encoded in the Clostridium perfringens genomes. we see that many of these strains also include important antibiotic resistance genes. And also we see a number of clades that have animal and human isolates. So, which is really important to say that animals may obviously be an important reservoir of Clostridium perfringens, and that's really important from an outbreak perspective and for tracking. And again, I think maybe slightly surprisingly, the Costa Rican isolates just themselves, they kind of fall all the way through the phylogenetic tree. So there's not really a strong regional signature here, which is really interesting. So we've got a load of stuff that we want to do. We've got a grant, we've got a joint grant that's under review at the moment. CIET-UCR, fingers crossed that we get it funded because it'll be the coolest project ever. But I just wanted to give you a little bit of insight into some of the work that we're doing with colleagues already in Costa Rica. And hopefully I can actually travel to Costa Rica, chat to CIET-UCR and the team, but some else of you will be really happy to chat about microbiome research that we're doing and new collaborations. And just the final thing I just wanted to highlight, and the beauty of using whole genome sequencing to look for kind of obviously different genomic signatures and link that back to clinical metadata, is actually with the work we've done with CIET-UCR. This is a preprint if you want to find out more, and we'll hopefully submit this relatively soon to the International Journal of Systematic and Evolutionary Microbiology, which is kind of the gold standard. We actually found a brand new species of Clostridium. So initially it looks very similar to Clostridium perfringens, but it's not. Genomically, it's far too distant, so we've actually named it Clostridium perfringinosum, which in Latin basically means similar but not quite the same. I think that's right, CIET-UCR. So I just want to say that's another really important thing about using whole genome sequencing, because we now think this is actually another important pathogen that nobody even had an idea was there. So we need to start looking at this as well. And last but absolutely not least, hopefully I think I've stuck to time, is acknowledgements. I'm just the PI, the group leader that sits in my office and basically responds to emails and then occasionally tries to help the team write manuscripts and also try to get funding to work together. But this has obviously been a huge important collaboration, because a lot of the work we do is translation with our phenomenal clinical colleagues. We could not do it without them. The fantastic teams, I've still got a team in Norwich, but we're growing now in Munich as well. So we will hopefully, when we can all come together, not to be socially distanced, I can update my photograph because this is an old one now. Our funders, obviously, as well. And obviously, huge thanks to Cesar, Carlos, and Cindy as well with respect to the new Costa Rica Clostridium perfringens projects we've got ongoing as well. And just last but not least, thank you very much for your time. And I'd be very happy to answer any questions. And I think we're going to do that by you submitting questions, and I will kind of write that back in a blog rather than a blob. And of course, very happy if there's anybody interested to contact me directly, then please do. And thank you again for your time.